Society Award in Pure Chemistry. Uh, Camille Dreyfus Teacher Scholar Award, and I could go on. It's a really impressive resume. He's been interested in what I like to, not his nomenclature, but what I'd like to think of as smart vesicles. In other words, vesicles that are self-assembling, propagating, and can respond to external stimuli. And he's going to talk, I hope, along these lines. And the title of his talk is Lipid Sponge Droplets as Programmable Synthetic Organelles. So Neil, thank you.
Yeah, well, thanks for that wonderful introduction, Peter. Everyone can hear me?
Yes.
Great. And yeah, I just want to, again, thank the organizers for the opportunity to speak today and share with you some of the recent work from our lab. I'm going to go ahead and share my screen here. Hopefully everyone can see that now.
I can see that.
Okay, great.
Yeah, that's it.
Okay, so I want to maybe start off with a sort of a very, very big picture, quite literally starting at the beginning, sort of motivating factor for our lab, like what really gets us going. And it's one of these really, really fundamental, I think, scientific questions. So if we look at sort of the timeline of the Earth, you know, it's thought the Earth was formed approximately 5 billion years ago. And actually, a billion years after the formation, unicellular life was formed. And then about a billion years after that, multicellular life is formed. And in fact, our own species, Homo sapiens, very, very short on the time scale, just only about 200,000 years ago. And so, you know, there's a lot of really good science sort of explaining, you know, how such a simple unicellular life eventually evolved to species as complex as our own. And there's a lot of really good hypotheses and theories that are backed up by a lot of evidence. But what really sort of fascinates us and many others is this very first step. How did you know, unicellular life appear in the absence of any pre-existing life? And so this is kind of, you know, literally the question of when did the chemistry become the biology? And so, you know, we find this question really quite fascinating. It's still in many ways a virtual black box. It obviously happened. And so our lab has become very interested in trying to develop approaches to try to gain some kind of better understanding over this, you know, what I feel is one of the most important problems today in chemical biology. Now, the definition of life is notoriously contentious. In fact, today there really isn't a universally accepted definition of what is life. But I think one thing that I believe the consensus opinion would be is that the minimal life is a single cell. And so for that reason, our lab has been interested in developing ways to create so-called synthetic cells or artificial cells. And these are, you know, typically structures that mimic either the form or function of living cells. Now, living cells are extraordinarily complex, particularly, you know, today's modern cells, lots of different functions, lots of different parts. It's a little bit daunting to say, okay, we're going to mimic everything that a, you know, current living cell does. And so for that reason, we decided to focus in on one particular structure of living cells that's extraordinarily important and universal, and those are lipid membranes. So lipid membranes, they're, you know, a hallmark of life. All living organisms have lipid membranes. They you know, act as containers. They define boundaries, which is important for enabling variation. They promote and protect internal chemical reactions, either the plasma membrane or for eukaryotic cells, the internal organelles. They control, exchange, and transport with the environment. And also, very importantly, they maintain ion and pH gradients, which are very important for example, energy production. Another thing I would just add is in many of the sort of theories about how life originated, which again is somewhat contentious, there's been sort of two camps: the camp that believes that metabolism came about first, and a camp that believes that genetic information in the form of RNA came about first. But what's interesting is, I think if you look at sort of the work from both of these camps, both will always invoke containers and compartments. And so I think that's one of the things I think drew us to lipid membranes, is that they are sort of the material that forms this container and compartment in cells. We thought it'd be interesting to explore them and mimic them. And just a little quick sort of primer or review of how sort of lipid membranes form. So cells are formed membranes from phospholipids, and these are molecules that have a polar head group and typically two acyl chains. And these will self-assemble spontaneously to form bilayers, which can close on themselves to form vesicles. Now, why is it that these molecules form bilayers? Well, a very simple explanation is it's just their shape. So if you think about that cross-section of the head group and the cross-section of the tails, they're approximately equivalent. And so this molecule has a shape similar to a cylinder. And these cylinders will naturally pack into these sort of planar arrays. Now, you can contrast that with a so-called lysophospholipid. So this is a phospholipid where one chain has been cleaved off, typically enzymatically. And now, because you've cleaved off one of these chains, the cross-section of the head is greater than that of the side chain. And so now it has, instead of a cylindrical shape, a conical shape. And this cone now is going to want to pack into what is known as a micelle, which is essentially kind of a monolayer that's folded in on itself. And so this doesn't have the appropriate shape now to pack into a bilayer. So how do people typically form sort of artificial membranes? Well, the very common way, and it's just done very routinely, is to take phospholipids that have already been formed synthetically or derived from natural sources and make a thin film of these phospholipids and hydrate them. And you will form, through various techniques, lipid membranes and vesicles. Now, typically the way we form this, these are very passive entities. So you may be forgiven for thinking that membranes and bilayers are very static compartments. They're not very interesting. They're not very dynamic. But in reality, in living systems, nothing can be further from the truth. So in a live cell, lipid membranes are extraordinarily dynamic. They're constantly being formed, degraded, involved in recycling processes, transport between organelles, for example. And so our lab became really interested, and I think this sort of hits a little bit on what Peter is saying, and I like the term, Peter, smart vesicles, smart membranes, that would be responsive to stimuli, that would be dynamic, that could mimic synthesis and degradation and remodeling. And through all this, sort of more closely mimic what happens in living cells and try to mimic these sort of dynamic membrane environments we find in live cells. And so one of the first things we were interested in trying to mimic is how lipid membranes are synthesized and formed. So obviously, in most labs, what's done is you just purchase phospholipids and hydrate them. Obviously, cells have to make their own phospholipids. And this is typically a really complex set of chemical reactions typified by the so-called de novo or Kennedy synthesis pathway, which is depicted here. So essentially, you build up fatty acids that are activated by the formation of thioesters. And then these thioester-activated fatty acids are coupled to phosphorus-containing head groups through the action of integral membrane proteins and forming phospholipids. And so this is obviously very complex. When we initially started our lab at UCSD, it seemed rather daunting to say, okay, let's mimic all the steps of this sort of so-called Kennedy pathway. And so instead, we became interested in trying to mimic another type of phospholipid synthesis pathway, and this is known as the so-called Land cycle or remodeling pathway. So in this Land cycle, what happens is there is a already formed phospholipids, and a phospholipase cleaves one of the acyl chains of a phospholipid. And that forms so-called lysophospholipid. Now it only has a single chain. And that lysophospholipid can combine through action of a lysophospholipid acyltransferase with a acyl-CoA, a thioester activated fatty acid. And so this acyltransferase will now couple these two components together, these two single chain components, and form a new phospholipid. And you may notice the tail has the potential to be remodeled. And that's why this is often referred to as the remodeling pathway. Now, what was interesting to us about this pathway is that you had a step here where the enzyme couples this lysophospholipid and thioester, a step where you take two single chain precursors that, if you recall, do not form membranes. These would form micelles typically, and couples them to form this diacyl product phospholipid that can form membranes. And so what we have here that's interesting is in a single coupling step, you go from non-membrane forming precursors to a membrane forming product. And so this to us was interesting. We thought, well, rather than trying to mimic all the complexity of the de novo Kennedy pathway, maybe we could mimic this simple coupling step now non-enzymatically to form phospholipids de novo. And so really the concept is simple. If a phospholipid is a head group and has two alkyl chains, maybe what we could do then is split this apart into two single chain precursors and use a variety of coupling chemistries to bring them together to form a membrane. And this could potentially happen in aqueous solution in situ, so you could form membranes de novo spontaneously. So, to do this, the very first example, we used a very, very robust chemistry known as click chemistry, or copper catalyzed azide alkyne cycloaddition. The reasons we chose this are simple. We wanted a chemistry that could work well in water because that's the solvent in which membranes self-assemble in. We also wanted it to be very chemoselective, robust, and we thought that this chemistry would satisfy all of these constraints. And so what we did was we made a lysolipid analog that contained an alkyne, which is shown here in red, and we made a long chain precursor containing an azide, here shown in blue. And in the presence of a copper 1 catalyst, these two functional groups will couple to form what is known as a triazole, which is depicted here, this 5-membered ring. And that links now these two chains together, forming an artificial phospholipid that's very, very similar to a natural phospholipid, except it contains this triazole functional group. And what's really fascinating is it turns out, when we initially did this, we had no idea, what was going to be the impact of having a triazole there. And surprisingly, there's very little impact in terms of the physical properties of the membranes that you form. And so you can measure things like the gel-liquid phase transition temperature, and it turns out they're very similar to that of natural phospholipids that don't have this triazole. Now, what's really quite interesting about this is now you can sort of do this sort of chemistry in situ, that is in water. And so here what I'm showing is some fluorescence microscopy in the presence of a membrane staining dye. And we've added these two azide and alkyne components. And so in the absence of copper, we don't see any membranes. But once we add copper, what we can see is these beautiful vesicles and tubules spontaneously emerging. These are all micron-sized structures. And with time, over 12 hours, you're left with fields of vesicles. And this is not just what we observe microscopically using, LCMS, mass spectrometry techniques. We can show that indeed the reaction is occurring over this 12-hour period where we're consuming the reagents. And so this concept has been repeated by our lab for other kinds of chemistries. Just highlight one that we find quite interesting. So we were interested in trying to more closely mimic what happens biochemically. And if you recall, biochemically, you make phospholipids typically from thioester precursors. And so we wondered, is there a way to non-enzymatically do this and using very selective chemistry? And so in the peptide or protein chemistry community, there's a very well-known reaction known as the native chemical ligation. And this is a way of joining two peptides together. And what you end up doing is you take one peptide and have an N-terminal cysteine, and you take another peptide and you modify the C-terminus to make a thioester. And it turns out these two will spontaneously react, forming a new thioester through transesterification. And this amine now is positioned so it can attack this thioester internally, essentially. And you have what's known as an S to N acyl rearrangement, irreversibly forming a peptide bond. And what this does then is obviously it joins peptide 1 and peptide 2 together irreversibly through the cysteine linkage. So what we thought to do, which is really kind of simple, is, well, could we just apply the same kind of chemistry, but instead of peptides, use it with lipids? So can we use this chemistry to tie together two lipid chains to form phospholipids? And so that's essentially what we did. A really great postdoc in my lab, Roberto Brea, who had a background in peptide chemistry, was able to work on this project. He created a lysolipid that has a cysteine, and then also created a thioester derivative of a fatty acid, oleic acid from oleic acid. And when you bring these two individually, neither of these form vesicles. In fact, both of these have charged head groups. So they form micelles. In fact, they're very easy to disperse in aqueous media. But when you mix them together, you spontaneously get this native chemical ligation and form a artificial phospholipid product. So again, very similar to a natural phospholipid, except you have this cysteine linkage. And so because neither of these precursors form vesicles, you can actually watch this process quite nicely. So here's a video I'm about to play where it's just using phase contrast microscopy. At the beginning, we don't really see much in the terms of tubes or vesicles, but as they start reacting, we can see spontaneously the formation of these sort of worm-like tubes. And these tubes grow as more and more reactions occurring, expanding the membrane. And with time, at the end, you're left with more spherical-like vesicles. And I often get asked this question, why do you see this sort of shape transition? You know, initially, you're making these long worm-like tubes. In the end, you have these sort of more spherical vesicles. So, you know, this is a very complex reaction mixture. And so at the end, we have phospholipids that want to form more spherical-like vesicles. In the beginning, we have only these single-chain precursors that want to form micelles. But in those intermediate steps, we have a mixture of the two. And it turns out that having a mixture of these sort of single-chain precursors and these phospholipids can lead to sort of these worm-like, highly curved structures. Okay, so over the years then, we have engaged in various projects that I think fall under this umbrella of cell mimetic membrane chemistry, or as Peter pointed out, sort of smart vesicles or smart membranes. So initially we started out with ways to non-enzymatically de novo form membranes, but we've also looked at self-reproduction of membranes. We've explored novel kinds of amphiphiles that can form membrane structures. We've looked at trying to mimic the remodeling process of membranes. Can we reversibly change the structure of synthetic membranes similar to how cells do? And then we've also started looking at more complex behavior. So if we make these artificial cells, can they communicate with one another? Can we actually start incorporating proteins and enzymes? So can we do integral membrane protein reconstitution using some of our membrane synthesis techniques? And more recently, even though at the very beginning, when I started at UCSD, we were interested in non-enzymatic ways to form membranes. More recently, we've become interested in asking the question, well, if we can use enzymes, what's the minimal reconstitution that we can do to form membranes from an enzymatic system? And we've been making some progress in that area as well. But of course, I think when we went into this area, as we started exploring these ways to create mimetic systems, we started realizing that there are a lot of other problems and questions that arise as we start doing this research. Can we achieve, for example, size and shape control of the membranes? Or can we try to better understand the role of membrane proteins? And so actually in the last few years, we've been mostly focused on three areas. So can we non-enzymatically form natural lipids? Can we use chemistry to manipulate living cell membranes themselves? But for the purpose of today's talk, I'm going to focus in on this project here, which is, can we actually utilize our synthetic membranes to mimic the function of organelles? And can we actually start creating and thinking about artificial organelles? And so that's what today I'm going to focus and talk about. So right up to now, I've been talking about phospholipid vesicles. Phospholipid vesicles are great. They're compatible with modern biochemistry. They're impermeable, so they can maintain ion gradients. But that impermeability can also be a detriment. So it's challenging for small molecules and proteins to traverse a synthetic membrane. And so that means if you want to get transport, you need to have sophisticated transport machinery. And of course, nature has solved this by evolving very, very sophisticated machines that can embed themselves in membranes and facilitate the transport of various molecules and proteins even across the membranes. But for creating simple cells and simple sort of primitive structures, this is kind of daunting to try to reproduce. Now, more recently, there's been a lot of interest in the community in what are known as membraneless organelles. So these are systems typically formed from polymers or proteins or RNA that are basically phase separated, a different type of phase, but still an aqueous-aqueous phase separation. And what's interesting about these sort of membraneless organelles, which are also found in living systems that can be made artificially, is that it's very straightforward for you to have transport in and out of the organelle. There's no actual membrane that can prevent the transport. But there are some detriments. And so one of the issues is that the membrane proteins themselves that we'd like to study obviously are not going to be able to be included in the organelle. And second, the programmability of these structures is also somewhat cumbersome. It's a little bit hard to, and still a very, I think, active area of research to figure out how do you reproducibly program the structures and what goes into the structures and what doesn't go in the structures. So recently our lab, as I kind of briefly mentioned, has been interested in exploring novel kinds of amphiphiles. And so in some work that came out a couple of years ago now, we showed that actually if you make simple single chain lipids from a galactose head group and an acyl chain derived from oleic acid, surprisingly, these molecules will self-assemble to form very beautiful cell-like vesicles. And cell-like meaning that they create these micron-sized structures that have bilayers. And so, this is quite interesting to us because these simple single-chain precursors were compatible with biochemistry. They're charge neutral, so they're not interfered by the presence of cations like magnesium and calcium, which have been problematic for other single-chain species that form membranes. And because they're single-chain species, we do see higher permeability. So very small molecules we found could go into and out of the vesicles formed from these amphiphiles. But they're still impermeable to larger molecules, such as proteins, for example, but even small oligomers. And there's a limitation in terms of the kind of surface area density you could have. So what we found quite serendipitously is we started exploring these galactolipids, if you will. And we, in the process of exploring these lipids, a grad student in my lab, Anja Bhattacharya, became interested in trying to reconstitute membrane proteins in these structures, in the membranes that are formed from these galactolipids. And in the process of exploring this, he started mixing various detergents with these galactolipids. And he noticed something quite remarkable. So if he mixed in a detergent known as IGPAL, which is this non-ionic detergent, instead of observing membranes or even micelles, he observed something quite different. He observed these really strange droplets, these very spherical structures. And these are definitely phase-separated droplets. So if you disperse them, they're in solution, but over time they will settle, or they can be centrifuged down to the bottom. So over 3 hours, in fact, they will settle and form a bulk phase. Now, when we initially did this, we thought, well, okay, we have these rather hydrophobic compounds. Probably these are just oil droplets. These are just droplets of hydrophobic species that are phase separated out. And Anja did some stability studies, found these were quite stable depending on the concentration of IGPAL and this galactolipid. But what was sort of really surprising to us is that when he tried to then expose these droplets to a fluorescently labeled protein that would bind to these head groups, sugar head groups, rather than seeing a ring around each droplet, which is what we would expect if the droplets were simply oil droplets, the entire droplet lit up. And so this really suggested that the protein was able to penetrate into the droplets, and that suggested that, in fact, the droplets had an aqueous interior. And so this sort of caused Anjit to embark on quite a bit of studies to try to characterize these droplets. We're not really a material science lab, but to Anjit's credit, he started collaborating with labs that have expertise in this area and really sort of taught himself quite a few different techniques. And so in collaboration with the physics lab at ECSD, the Sunil Sinha lab, he was able to do various types of X-ray studies on these droplets. He was also able to do various types of high-resolution microscopy like cryo-SEM. And to sort of make a long story short, what we concluded from all this characterization work was that rather than these droplets being oil droplets, what we have is actually another sort of liquid-liquid phase separation where the interior of these droplets are actually what are known as a L3 bicontinuous sponge phase. Okay, so what does that mean? That's kind of a mouthful. It means that inside these droplets, there's actually a three-dimensional network of lipid bilayers. And it's both the aqueous and lipid domains are continuous through this droplet. So again, as the name sort of implies, sponge phase, one of the ways to think about the interior is it sort of looks like a sponge, where both the aqueous and lipid domains are continuous throughout the sponge. And so this means you have an extremely large surface area of lipid membranes. By measurements through the X-ray spectroscopy, we were able to estimate the water channels are about 4 nanometers in diameter. And about 67% of these droplets are actually with just water. So in fact, they're quite laden with water. There is a very aqueous interior. This is another sort of picture, cartoon that might sort of better illustrate what we believe is occurring inside these droplets. So like I said, it's sort of a sponge-like interior. These sort of blue structures are the membranes, and the white space would then be aqueous channels. So it turns out that these structures can be formed by a few different types of detergents. We're still trying to explore the range, and this is something that's of active interest to us now. But we can change, for example, the side chain length of these galactose sugars, galactolipids. We can change the detergent to other types of non-ionic detergent, but though very similar to IGPAL, like Triton X100 or Triton X114. And in all cases, we can observe droplets. We do observe sometimes changes in the average droplet diameter, but this is something that I think we need to do more to reproducibly understand how the structure of the detergent precursors actually affects the structure of the droplet and size of the droplets that are formed. Nevertheless, we were really interested to see, could we start exploring applications of these droplets? And one of the first questions we asked was, well, how stable are these droplets? They're made from this mixture of detergents. Would they be stable to various types of conditions of pH, cations, et cetera, et cetera? And turns out they actually are quite stable. So, you can change the pH from 2 to 10 and the droplets persist. We can add high concentrations of salts, 1 molar sodium chloride, for example, magnesium, calcium, the droplets are fine. Even seawater is fine. Cell culture media, so DMEM FBS, which is what you grow tissue culture cells in, it's also fine. And then some of you may be aware of the PURE system. This is actually a system that enables you to use recombinant proteins and molecules to produce proteins in vitro. And in fact, in that PURE system mixture, the droplets are also fine. They're quite stable. So with that in mind, we became interested in asking how do various types of molecules partition and behave in the presence of these droplets. And so one of the easiest ones to test, or classes of molecules to test, are fluorescent dyes. And so we basically expose these droplets to a wide variety of commercially available fluorescent dyes. And to make a long story short, basically, if the dye is more hydrophobic, we could see it incorporate or partition more so into the droplet than if it's more hydrophilic. And a good example of this is actually fluorescein. So fluorescein has a pH dependency on its charge state. And so if we change the pH and create a more hydrophilic fluorescein through deprotonation, we see very little incorporated in the droplets. But when we then lower the pH so that we protonate the fluorescein, we actually get much more incorporation into the droplets. Now, perhaps more interesting than small molecules like fluorophores are proteins. So as I mentioned, the channels are actually quite large, and so we were curious whether or not proteins would go in and out of these membranes, and how would that behave. And so what we can do is, because we have these detergents, these galactolipids that have galactose head groups, we can take proteins that are galactophilic, like lectins, LAC-I, galectin-3. These all have a tendency to bind to sugars and galactose. And we can fuse them or modify them with either fluorescent proteins, fluorescent molecules, and observe how they partition in the droplets. And what we found was kind of quite remarkable is these droplets, kind of like a sponge, have the ability to soak up these proteins from the environment and really concentrate them up. So for just an example, if we take lectin, LEC-A, and have that in the presence of the droplets, we find that 97% of the material gets sucked into the droplets. In fact, we can see a concentration increase in the droplets of almost 100-fold. So we can achieve now, concentrations of proteins in the droplets up to like 100 micromolar or even more, actually. And so I think the droplets turn out to be a really great way of creating a very protein-rich environment. Now, one kind of nice way to observe this is, in fact, we can, as I mentioned, the droplets are stable in the presence of the so-called pure system that allows you to do in vitro transcription translation. So you can have droplets now in this in vitro transcription translation system and add DNA that encodes for a GFP fusion to LEC-A. And when you do that, you can actually watch then protein synthesis occur. And this is a video I'm going to show you now, so you can see all these droplets are present. And we've added the DNA for GFP LEC-A. And we observe is, in fact, you don't even see the GFP LEC-A outside. They immediately get sucked into the droplets. So this is real-time protein expression and immediate shuttling of the GFP LEC-A into the droplets. So, so one approach then in terms of programming what goes into the droplets is to fuse the protein of interest with a galactophilic protein. That's a little bit cumbersome. If we take GFP alone, well, GFP alone doesn't spontaneously partition into the droplets. So we started thinking about, well, what would be some other ways in which we could try to program what goes into the droplets? And so one idea was, since the droplets consist of this sort of three-dimensional network of lipid membranes, could we dope in functional amphiphiles at a low percentage and use that as a way to determine what goes in or out of the droplets? So what do I mean by that? So an example would be, we sort of, as one of our first examples, we doped in a small percentage of a phospholipid that has a nickel NTA binding head group. And so this is now a head group where a polyhistag protein in the presence of nickel would want to bind to this phospholipid. So if we don't have this phospholipid and we expose now histag GFP to these droplets, we don't see anything go in. But if we add a small percentage of this phospholipid into the droplets, now a polyhistag GFP will be enriched quite a bit in the droplets. You can see all the droplets have split up. And so this can be repeated through a number of other sort of variants. So we can put in other types of sugars, like a mannose sugar, dope that in. Now mannose binding lectins will go in. We can put a positively charged amphiphile. Now negatively charged molecules like oligonucleotides will go in. And then, again, very, very convenient for biotech applications. You can put a biotinylated phospholipid inside these droplets, and now streptavidin will be enriched very highly in the droplets. And so this turns out to be a really nice way in which you can very reproducibly program what goes into the droplets and what doesn't. So at that point, we became interested in asking whether or not we could utilize these droplets and try to mimic some of the functions of organelles. And so in eukaryotic systems, there are these membrane structures inside the cells. Many of them are highly networked, like the smooth endoplasmic reticulum. And so we thought that our sort of highly networked three-dimensional droplet structures could try to mimic some aspects of organelle function. So for instance, could we mimic lipid synthesis that occurs in the ER? Could we mimic protein degradation that occurs in lysosomes? Or could we use our droplets to sequester and protect biomolecules, which occurs in stress granules and in nucleolar detention phenomena? So before doing that, one of the first questions we had was, well, what do molecules do when they're entrapped in the droplets? Are they static, or are they moving around like a liquid? We notice that these droplets can fuse with one another. So that's this video playing here. There's 3 droplets fusing into one. This sort of suggested to us that, in fact, these droplets are very liquid-like. But Henrike Niederholtmeier, who became very interested in this project, wanted to sort of more quantitate this a bit more. And so she did these fluorescence recovery after photobleaching experiments to try to understand how dynamic the interior of these droplets are. And so this is a very simple set of experiments where we put fluorescent compounds in the droplets, small molecules like a Texas red lipid or a protein. This is a fluorescent protein, mCherry his-tagged, program their entrapment into the droplets, and then use a laser to burn essentially a hole, photo bleach a hole into the droplet structure, and then watch with time, 1, does that hole recover, and 2, on what time scale. If the droplets are completely static and have static interiors, this hole won't recover. But as you can see, after you make the hole, within a minute or so, you have recovery. And so from that, you can make at least an estimate of sort of how diffusion is occurring in these droplets. The diffusion is certainly less so than in, simple bulk solution, but it's fast enough that we felt there was mobility so that molecules that you entrap in the droplets would be able to interact with one another. And so this, again, gave us confidence to start exploring various kinds of applications where we could entrap various proteins or molecules in the droplets and look for chemical reactions. So one of the ideas we had was whether or not we could try to think about entrapping membrane proteins in these droplets, because we have this 3-dimensional network of lipid bilayers, and this is something that we could study that really can't be done very easily with other kinds of droplet systems. And so it's really quite simple to reconstitute a membrane protein in this system. Basically, you can dissolve a membrane protein in the detergent, either IGPAL or Trinex, and then simply add the galactolipid. And then you spontaneously form these droplets that incorporate the membrane protein in the bilayers. And so our very first example of this is a very common membrane protein, cytochrome C oxidase, terminal protein in aerobic respiration, oxidizes ferrocytochrome C. So we could reconstitute cytochrome C oxidase in the droplets. We've fluorescently tagged it so you can see it. It's now quite enriched in the droplets. And when you add ferrocytochrome C, it gets rapidly oxidized by these droplets. And so that indicates that the enzyme is still very much functional even within the droplets. As I mentioned, one of the things we were interested in mimicking was lipid synthesis. And so we were curious if we could reconstitute a membrane protein that would react with lipids. And so for this, we reconstituted diacylglycerol kinase. So it's a membrane protein involved in lipid modification. And then we could take a lipid substrate of diacylglycerol kinase and show that in the presence of magnesium and ATP, indeed, the droplets were able to phosphorylate this molecule. And so I think what this suggests now for future work is whether or not we could try to elaborate on this further. Can we actually reconstitute a lipid synthesis pathway in these droplets? I think these droplets are also, because they are able to bind to membrane proteins and bind to hydrophobic small molecules, they might be a really privileged environment to have sort of lipid modifications, lipid chemical reactions occurring. So another question we had was whether or not, if we're able to achieve these sort of higher concentrations, localized concentrations of molecules and proteins in these droplets, does that sort of higher localization benefit reaction rates between molecules and proteins. And so to look at this, we decided to see whether or not protein degradation or peptide degradation would benefit from colocalization in the droplets. And so to do this, we took a protease and his-tagged the protease and used droplets now that can bind to this his-tagged protein. And this is cathepsin K. So we took then a peptide substrate for cathepsin K that actually has a coumarin product. This is a fluorophore. So if you have cleavage by cathepsin K, you release a fluorophore that turns on in fluorescence and gives you a really easy readout for whether or not you have protease reaction. And so you can mix these two together at low concentrations. There's just 2.3 nanomolar enzyme and peptide substrate, only 80 micromolar about. And so in the absence of the droplets, because there's very low concentrations, we actually don't see much degradation. And if we add detergents, we see some slight increase in degradation. But now if we have the droplets, where we have this sort of co-localization of the components, we see a really big boost in degradation rate. And so again, it suggests that we can use our droplets to study, actually it's a really kind of fundamental question, which is what's sort of the effect of scaffolding or sequestration of proteins and molecules on biochemical reaction rates. I mean, cells go through a lot of effort to organize chemical reactions, organize structures. And so I think it's been a question of, well, what's the impact of that organization? And so our system, because of its programmability, gives us a nice platform to study these kinds of questions. And we're right now collaborating with some groups to look at this more closely with various kinds of systems. Okay, so finally, we were interested in gaining more control then. So I showed you that we can program the localization of various molecules and proteins into these droplets. But we were also interested in gaining spatiotemporal control over what and when proteins in particular get partitioned into these droplets. And so for spatiotemporal control, really the best tool is light. And so we were really interested in asking, can we develop a way to use light to trigger whether a particular protein goes in or out of the droplets? So to do this, we decided to take advantage of a well-known pair of proteins derived from plants that are known to interact with each other reversibly based on a light source. And these are the so-called sort of optogenetic tools. And so the pair of proteins we worked with are known as Phi-B, or photoreceptor phytochrome B, and phytochrome interacting factors, or PIF. So these proteins, when you shine 660 nanometer light on them, Phi-B undergoes a conformational change and will bind now tightly with PIF. And in the if you add now instead 740 nanometer light, this will be reversible, and the PIF will no longer bind to the Phi-B. And so what this really means is you can use now light of two different wavelengths to reversibly go back and forth between this interacting and non-interacting state. So the way we wanted to then implement this is to take the droplets and immobilize in the droplets using the streptavidin programmability Phi-B. And we took PIF and infused it to GFP and put that into solution. So now if we shine 740 nanometer light, these two won't interact with one another. But if we shine 660 nanometer light, the PIF GFP is going to want to interact with the Phi-B, and that should be driving the PIF GFP into the droplets, and the droplets should light up. So that was the idea. And so here I'm going to show you a video of how we implemented this. So just to give you some perspective here, so here's the time on top left. Here's the wavelength of light we're shining. So at the beginning, it's 740 nanometer light. So PIF and Phi-B are separated. You can see the droplets here somewhat. So these are those structures I'm sort of highlighting with my cursor. Now I'm going to play the video. So we switch on 660 nanometer light. Now you can see the PIF GFP rapidly get enriched in the droplets. We hit it with 740 nanometer light, it rapidly goes out. 660 nanometer light goes back in, and this can be basically done over and over and over again. So it's very reversible. Okay, so how then do we sort of maybe apply this? So one of the things we're interested in asking is, if we drive proteins into the droplets, does that help protect them in a way by sort of detaining them in the droplets and preventing them from interacting with molecules or enzymes that can't go in the droplets? So to sort of look at this, we decided to add a tag, the so-called SSRA tag, onto this GFP-PIF fusion. And this SSRA tag makes this protein now recognizable by this degradation machinery known as ClipXP. The ClipXP is this large 600 kilodalton protein degradation machinery that will degrade these various tagged proteins. But importantly, because of its size, it's thought to be greater than 10 nanometers in size, we would not expect the ClipXP to go into the droplets, because if you recall, the droplets have these 4 nanometer pores. And indeed, if we take ClipP even, that's fused to GFP, and add it to the droplets, we can't even drive it into the droplets. You can see, we just see the surface of the droplets light up. The ClipP isn't able to get in. So now what we can do is we can mix all of this together and see sort of how does protein degradation respond to light wave. So if we use the just the 740 nanometer light, where the PIF and the Phi B are separated, we see this really nice degradation curve. So clearly the ClipXP is degrading very rapidly the GFP. But now if we intersperse in 660 nanometer light at these time points, when we add that, when we now expose these droplets to 660 nanometer lights, we see this really quite a significant slowdown in the rate of degradation, as indicated by the steepness of this degradation curve. So by pushing the PIF GFP into the droplets with that 660 nanometer light, we're able to detain the protein and greatly lower the rate of degradation, presumably because we're sequestering the protein away from the ClipXP. And so this is something that we're further exploring, not only to see whether or not we can push protect the protein by putting in the droplet, but can we actually drive proteases, kind of like I showed you previously with the peptide degradation, can we drive proteases as well into the droplets to speed up degradation? So we can start viewing these droplets as sort of organelles where you could programmably degrade various types of substrates. So with that, I'll just wrap up and maybe just state a little bit that our lab has over the years really gotten into this area of lipid membranes, dynamic lipid membranes, smart vesicles, really for basic science questions. We were really initially interested in questions related to the origin of life, and we've become more interested in very fundamental questions around lipid self-assembly. But I think more recently, in the last few years, by sort of taking the lessons that we've learned from the basic science, we've been able to apply our systems to various problems in biomedical science and biology. So I didn't have really time to talk about it, but we've been utilizing our tools to try to have a better understanding of the function of lipids in living cells, or to try to create therapeutics or diagnostics that can manipulate lipid synthesis pathways or lipid modification pathways in biology. And I think these lipid droplets are another great example of this. We've really up to now been very focused on the basic science. How do these lipid droplets self-assemble? Can we mimic organelles? But more recently in our lab, we've begun trying to utilize these droplets as ways to potentially reconstitute membrane proteins, or even try to create unique kinds of sensors, for example. And that's something that hopefully we'll be able to report on in the near future. And so I think it's also kind of a nice testament to, the importance of basic science. We wouldn't have gotten here with these applications had we not initially started here on these really basic questions. And so I'll just acknowledge our various sources of funding. I think we've been really fortunate to be funded by a variety of sources over the years. Particularly the work I talked about today was funded through the NSF EAGER program, as well as the Department of Defense Muri Award, and for separate aspects of what I was talking about today. And also, I would like to thank our collaborators and also all of our group members. Really sort of been a really great team of scientists that are really interested in this sort of boundary between chemistry and biology and interdisciplinary science. And again, I want to thank the organizers for the invitation, and I'd be happy to answer any questions.
Awesome. Great talk, Neil. So questions are coming in and I'm gathering those, but to start us off, I had a quick question. If you had two population of your lipid droplets, say one with one membrane protein and another with a second, and you mix those together, will they actually come together? And you could almost stepwise build organelles by introducing different proteins. And you can imagine kind of doing reverse knockout experiments with that kind of system.
So you mean if you have a one droplet with protein A and another droplet with
Yes.
protein B, could you mix them, and would the result have a mixture of protein A and B?
Yes. Or do you see localization to maybe half the droplet?
So right now, what we've seen is complete mixing.
Okay.
But I think what you're hinting at is the idea of like having localization. I think it's potentially achievable, but right now, absolutely, you can take a population of droplets that have protein A and a population of droplets that have protein B and mix them together, and you will get A and B to co-localize upon droplet fusion.
Mm-hmm.
Now, we have noticed sometimes phenomenon where it seems like there's sub-localization of various kinds of proteins within a droplet. And that's something that we're trying to explore now. Actually, it's part of this MURI award, which was recently awarded for, we're interested in trying to explore whether or not we can use these droplets as a way to hierarchically assemble proteins and other kinds of molecules within a particular droplet. So can we have layering, for example? Could the interior be different from the exterior? Can we decorate the droplets with different kinds of proteins? So that as you go from the inside to the outside, you have a sort of a change in the sort of chemical environment. And so I think that's something that we're right now really interested in exploring.
Especially to mimic, cytoplasm versus nucleus, potentially.
Yes, yeah, absolutely.
All right, we have a question from Tom Thompson. He asks, have you ever mixed the droplets with apolipoproteins, such as ApoA1, which can form protein lipid discs?
No, we haven't done that. So that's a really good question. We have actually been interested in trying to take our synthetic lipid membranes and mix them with apolipoprotein to see if we could form sort of de novo nanodiscs. And that we didn't really try too much on that, but that was something that we were interested. We weren't able to see results that particular time, but we were kind of interested in that question. But we haven't tried taking apolipoprotein and mixing them with these droplets. So that's actually a really, really good idea.
We have another question from Peter Stambrook. He asks, if you introduce lipid droplets into cells, say for Xenobus eggs, how stable are the droplets? Can they interact with cellular organelles?
Yeah, so that's a good question. So we've done we did a little bit of work trying to see, I mentioned the droplets are stable in cell culture media. We did a little bit of work trying to add the droplets to mammalian cells in culture. And there we actually, at the time, noticed quite a bit of cytotoxicity. So we're not too sure if what was happening is the droplets were coming apart. There's a lot of detergent in the droplets, so we don't know if the detergent was having an effect. We did notice some really strange phenomena in terms of how the membranes of the cells were affected by the presence of these droplets. But at the time, we really didn't have somebody that was interested in exploring this interface of, it's funny, sometimes when you're working with, in this group, you have people that are interested in the basic science and interested in sort of translational aspects, but then finding someone that wants to do sort of the in between can be sometimes tricky. So we haven't really pushed on this to see, okay, is this toxicity reproducible? Are there ways to get around the toxicity by making the droplets more stable? So that's something that actually we're really interested in, is can we stabilize these droplets either through cross-linking or adding various kinds of proteins so that they don't sort of, leak detergent or, or even fuse for that matter, So I think that's something that we're gonna have to look into more in the future. As of now, the only data I have is it seems like the initial droplets we made are somewhat toxic to cells.
We got another question. and I apologize if I mispronounce this, from Uche, but he says, my question is, what is the possibility of using CHIP or CIP-XP technique to prevent Alzheimer's disease by slowing down the degradation of amyloid beta precursor proteins? Could this be a new niche yet to be created?
Sorry, man, can you maybe repeat that question one more time?
Yeah. He says, what is the possibility of using CIP-XP technique or CIP-XP technique to prevent Alzheimer's disease by slowing down the degradation of amyloid beta precursor proteins?
Okay, I think maybe he's referring to CLP, like the clip XP. So I think maybe if I'm understanding this correctly, yes. I think it's saying that, can we basically utilize this as a way to programmably degrade proteins in a living system? And so, I think it's a possibility. I think the things we'll have to overcome are, like I just pointed out, the stability of the droplets to make sure they're stable in these kinds of environments. Like for example, in a human or something like that, which we're really not there yet right now. But we have thought about ways in which we could try to, in general, utilize these droplets as a way of sequestering, for example, toxic proteins. Like one thing we were curious about that we really haven't explored is, for instance, LEC-A is actually a biofilm virulence factor that's important for infection. So can we actually use the droplets to soak up LEC-A, and would that have sort of a therapeutic benefit? So these are kind of the ideas that, could be potentially explored. But I do think there are obviously big hurdles before we can get there, in terms of the, the compatibility, the biocompatibility of the droplets, for example.
We had another question from Peter Stambrook. He asks, have you tried to introduce ion channels into the vesicle membranes?
No, not yet. The vesicle membranes. Yes, in the vesicle membranes, yes, we have. We in our very first work, we introduced a type of ion channel and made actually membranes across the nanopore, and then measured ion currents and showed that these ion channels were functional. So yes, it is definitely feasible to introduce ion channels in those structures.
I had another quick question about the vesicles. If you let the reaction go consistently, is there an upper level to how big these vesicles can get to, or does it kind of level out?
That's a good question. So one of the things is these vesicles, they're very heterogeneous populations. So it's kind of hard to say, is there like a critical size that we see upon which, they burst or something like that? Right now, the answer is it's such a sort of a mixture of sizes that it's hard to really say that. We routinely will see vesicles that are over 10 microns in size. So, it's very feasible to have very large vesicles, and they don't seem to burst. But, that being said, it's not like we see 100 micron size vesicles. But I don't know if that's just simply because, they're being agitated, and then that's the mechanical forces are disrupting them. I think that's something that, I kind of hinted at. One of the things that we're interested in is size control of these vesicles as they're formed. And we think that we have some ways to get to that. And I think if we can start controlling the size and getting more uniformly sized populations, we can start asking questions like that more sort of reproducibly and reliably. Like we could try to, for example, grow a population, that's more homogeneous in terms of its size, and see where does it level off, or how does the environment or reaction affect the size.
So I think we've pretty much run out of time. I want to thank, first of all, Neil, for a really fantastic, fascinating, provoking talk. Certainly has me thinking, which is rare. And Eric, thank you for handling the Q&A, the questions. And of course, Jessica, for making sure this worked seamlessly. So thank you all for your attention. Appreciate it, and hope you'll tune in on the 25th when we have another session. And any other questions? No. Okay. I am going to check out. Thank you.
Thank you all.
Thank you.